The sodium-potassium pump is crucial in creating the concentration gradients of sodium and potassium ions across the plasma membrane in most animal organisms. This enzyme transports 3 sodium ions out of the cell and transports 2 potassium ions into the cell. The enzyme uses the energy from the hydrolysis of ATP to go through its conformational changes. The sodium-potassium pump is crucial in neuron signaling because it allows for action potentials to be used to communicate with other neurons. The sodium-potassium ATPase has 3 subunits. The alpha subunit has 4 domains: an actuator domain, colored in yellow, a nucleotide binding domain that interacts with ATP, colored in red, the phosphorylation domain that interacts with the ATP to go through autophosphorylation, colored in blue, and a 10-helix transmembrane domain, colored in gray and black. The beta subunit, colored in purple, is crucial for sodium-potassium ATPase membrane stability. And lastly, the FXYD domain, colored in cyan, is responsible for regulating the enzyme's binding affinities for sodium, potassium, and ATP. This second diagram shows the calcium pump, but the mechanism is the same since the beta subunit is not involved with the activity. The enzyme follows a cyclic mechanism between the cytoplasmic open E1 state and the exoplasmic open E2 state. First, in the E1 cytoplasmic open state, the enzyme binds sodium ions and binds ATP to the active site, forming an E1 ATP conformation. When ATP undergoes autophosphorylation, the enzyme switches to the E1P ADP sodium occluded state. This state is not open to the cytoplasm anymore. Gradually, this enzyme switches to the E2P state that is now open to the exoplasm. This releases sodium ions outside of the cell and binds 2 potassium ions. This release of bound phosphate from the E2P state causes a conformational change to the E2 potassium occluded state. The enzyme then switches back to the E1 state and then releases potassium ions into the cytoplasm, ready to start the cycle anew. In this first scene, the enzyme is in the sodium and ATP bound E1 state. The substrate used to characterize this conformation is AMP-PCP, which is an ATP analog. The alpha subunit is in yellow, the beta subunit in white, and the FXYD subunit in light orange. The nitrogen atoms are colored in blue, the oxygen atoms in red, the phosphorus atoms in orange, and for substrates, the carbon is colored in green. The active site shows an AMP-PCP molecule in place of ATP, which is in the P domain of the alpha subunit. ACP2001 stands for AMP-PCP. The P domain contains an aspartate residue, which is shown in the model, which acts as a phosphoacceptor and is part of the universally conserved DKTG motif. The lysine and threonine of this said motif are also shown. The ATP has not yet been hydrolyzed at this stage, so the aspartate is not attached to a phosphate group. The aspartate residue of the DKTG motif interacts with the gamma phosphate of ATP. In the N domain, the adenine of ATP binds in a pocket and interacts with the phenylalanine residue, and there's also a magnesium ion that interacts with the phosphates of ATP, but it is not characterized in this structure. Here, the E1 ATP conformation of the protein is shown in yellow as before, and the E1P ADP conformation of the protein is shown in green. The individual atoms are also colored the same, with nitrogen, oxygen, and phosphorus corresponding to blue, red, and orange respectively. The green enzyme represents the conformation where ATP has been hydrolyzed and is the intermediate between the ATPase switching to the E2 state. When ATP is hydrolyzed into phosphate and ADP, the interdomain linkage is broken, causing the aspartate residue to move slightly to accommodate the E2 exoplasmic open state. Also, after the interdomain linkage is broken, the A domain of the alpha subunit rotates roughly 120 degrees. This is coupled with the exoplasmic gate opening and the transformation to the E2 state. We can see here the E1P ATP conformation clearly when we remove the E1 ATP conformation. After the interdomain linkage is broken, the aspartate still interacts with the phosphate while the phenylalanine residue interacts with ADP. The phosphate is represented as aluminum tetrafluoride, colored gray for aluminum and cyan for fluorine. We can see that the aluminum tetrafluoride interacts with the aspartate 366 residue. The aluminum tetrafluoride is the stand-in for the phosphate group. The transition between these two conformations describes the hydrolysis of ATP needed to drive the cycle forward for sodium-potassium pumps. Once the ADP is released into the cytoplasm, the enzyme switches to the E2 state, where it can then release sodium out of the cell and take in potassium ions.